Alright, so today we're going to be talking about a developmental biology topic, the formation of somites, and how the 90-minute clock works. So first, let's kind of go over what somites actually are. So somites are actually these bilateral pairs of mesoderm, and they run along the anterior to posterior axes. And in this diagram here, you can see there's a neurotube running down the middle, and we have bilateral pairs on both sides here. So eventually, somites will diverge and form different types of sclerotome, myotome, and dermatome. So the sclerotome will contribute to the vertebrae and the ribcage, myotome into skeletal muscle, and dermatome into the dermis or the skin. So the formation of the somites is really the very beginning of the formation of the rest of the body plan, like these tissues here, these tissues and bones that we see here. Okay. And so when you're forming somites, there's lots of signaling levels that are quite crucial in how that somite and when that somite will be formed. So the two that are really quite important are FGF right here and retinoic acid, or RA for short. And so these are actually secreted at opposite ends, and they're actually antagonistic to each other. And so antagonistic would mean that FGF is suppressed by retinoic acid, and retinoic acid is suppressed by FGF. So at the formation of somites at the wavefront, somites are actually formed at a specific low threshold level of FGF. So it doesn't matter if you have a really high level of FGF or basically no FGF. You need that very specific low level of FGF for your somites to form. Okay? And so we've been talking about the 90-minute clock in the first slide of this, and you've probably heard it before in lecture. But the 90-minute clock is specific to the chick embryo. And what that means is that almost exactly every 90 minutes, there will be a new pair of somites forming. And with each pair of somites forming is sort of a new wavefront cycle. And this 90-minute clock is actually due to negative feedback loops, beginning with FGF. So let's go over the signaling pathway here. So first we have FGF, which I had gone over in previous slides. Okay, and FGF is going to activate Wnt3a. Wnt3a is going to activate Notch, which will activate Hairy1. Okay, which will then activate Effrin A4, and Effrin A4 is what causes the segmentation and formation of somites. All right. So far we're only looking at the very top part of this signaling cascade here. But Effrin A4 is really what causes that final formation of these somites here. So this is all initiated by FGF. So how does this negative feedback loop work? How does it play into the 90-minute clock? Well, when you're activating Wnt3a by the presence of FGF, you're also activating axin. And axin will in turn come back around and repress Wnt3a. Okay? So no more Wnt3a is being made. Okay? So a negative feedback loop is when the output of a system will actually turn off that system itself. All right? And so when you're getting the repression of Wnt3a, you're shutting off this part of the pathway here, and there's no more Wnt3a. Well, if there's no more Wnt3a, then also Notch can't be activated. But when Notch is activated, before Wnt3a gets shut off by axin, Notch will actually activate MESP2 in addition to Hairy1. Okay? So then MESP2 will then activate L fringe, which will then in turn come back around and inhibit Notch. So Notch is getting inhibited, but Notch has already activated Hairy1. All right? So before it gets inhibited, it's already activated Hairy1, and then this negative feedback loop comes around and turns it off again. And so you get this kind of cycling of when 3A gets turned off, and then FGF is turning it back on again, and it comes back on again, okay, and it turns on notch, and then Harry, and then F1 A4, and then it turns back off again, and then it turns back on again, and that's how the 90-minute clock works. It's due to these negative feedback loops. If we didn't have these negative feedback loops, we wouldn't have this 90-minute wavefront cycle at all, all right? Okay, so we just want to go a little bit more into how the wavefront works. So the wavefront is going to keep moving posteriorly, so this would be an anterior part, and here would be posterior, so anterior here, posterior here, and so because that wavefront is moving posteriorly, and it starts at the anterior part, cells that are more anterior are going to experience less expression wave cycles than cells at the posterior end, okay? So a good example of that is we can see this red dot here that could be representative of a mesoderm that is destined to be a somite. So it will go through several more cycles until it gets defined to be a somite here, so after 18 hours. Whereas if you had a cell that was up here, more at the anterior level, closer to the anterior end, after just maybe 1 or 2 cycles, it's already been defined as a somite. It's already been formed. Okay, so cells at the posterior end will definitely experience many more expression wave cycles than cells at that anterior end due to the wavefront cycling, okay? And so another thing to keep in mind would be that pre-somatic mesoderm is already determined prior to that formation and segmentation of somites. And we can see in this textbook example here, so we have a pre-somatic mesoderm here, and that's from a thoracic vertebrae, okay? That's going to be transplanted into a different embryo, okay? So a younger embryo here is a stage 7 embryo, and this is a stage 10 embryo. And you can see we're transplanting it from different regions. So here would be a thoracic region, okay? And then here we're transplanting it into the cervical region, all right? And so these are the results of the experiment. So in this region here, instead of just having regular cervical vertebrae, we actually have ribs forming, all right? And that's due to the fact that these mesoderms, they already have their identity predetermined. If this wasn't the case, and somite identity was fluid, we might expect the cells to dedifferentiate, and then based on the environment, based on the new environment that they're in, they would take up an identity that was appropriate of that new environment. So in this case, if a somite identity was not predetermined, we would see just regular cervical vertebrae here, and the chick would be relatively normal instead of having a bunch of extra ribs here from the thoracic region, all right? And so another thing to go over is the combinatorial gene expression of how different tissues will be formed, okay? So a really good example of this would be the ABC flower model. It's quite simple, so that's what I'm going to go over first very quickly. So combinatorial gene expression basically just means that each combination of genes will specify a different identity along its axes, okay? So in the ABC flower model, we have another video on that as well, so I'll link that below. But we're dealing with 3 classes of genes here, so A, B, and C. And if you only have B being expressed, you'll form sepals, okay? If you have A and B expressed, you'll form petals. If you have A and C expressed, you'll form the stamen. And if you just have C being expressed, you'll form the carpel. But all of these structures are formed using a combination of these same 3 genes, okay? It just depends on what is on and what is off, all right? So that would be a very simple example of combinatorial gene expression. So here we're going to get a little bit more complex. We're going to be looking at Hox genes. Hox genes also have combinatorial gene expression similar to the ABC flower model. Here we can go over another example of how combinatorial gene expression can define different regions. So if genes 1, 2, 3, and 4 are expressed, you'll get tissue Z. If you only have genes 1, 2, and 3 expressed, you might see tissue Y. If you have genes 1 and 2 expressed, you'll see gene X, or tissue X, sorry. And if you only have gene 1 expressed, then you'll only get tissue W, okay? But all of these tissues are expressed via these 4 genes, these same 4 genes, okay? So with Hox genes, it works in the same way, but there are just so many copies of them, and there's lots of redundancy in Hox genes, so it becomes very complex. But you could have A1 expressed and B1 expressed, and then D1 expressed, and see like what does that code for. So it works in relatively the same way as the ABC flower model in this simple example that we went over here, but because of the redundancy and the complexity of it, we won't be going into it too much. So I have a few questions here on Somites, and it would be really good for you to just test your understanding. So maybe give these questions a read through, and if you don't understand them, go back through the video and try to see where you have a knowledge gap, okay? So I'll leave this slide up for a little bit, but otherwise, thank you guys so much for watching, and hopefully we'll see you in the next video.